thanks everyone for taking the time to come and listen about Avacta and the other companies that are presenting. So the presentation, by the way, is downloadable from our website as well under the investor section if you want to get a copy of it. So I'll start by giving a very quick introduction, a scene setter, if you like. So antibodies, and we'll talk through all this in a bit more detail through the presentation, but antibodies have come to dominate some very big markets, hundreds of billions of dollars worth of market through research tools to diagnostics through to therapeutics themselves. And that's despite some fairly significant limitations. And what we've developed in Avacta is an engineered alternative to antibodies that doesn't have those types of limitations. So we're now commercializing, as you can see, in the non-therapeutic space. So this is providing Affirmers to diagnostic developers, for example, and we'll talk through that in the presentation. And we also have our own pipeline of therapeutic Affirmers that we're developing in oncology. So that, hopefully, I'll give you a feel as we go through the presentation that we can really influence the development of medicines right across that piece, from the early research providing powerful tools through diagnostics through to the therapeutics themselves. That's what I hope I'll get across. So antibodies then, for those who aren't familiar with what an antibody is, I think what's important to understand is the reason they have some commercial value is that an antibody can bind to a target. It can identify the target and bind to that target. And then that can be used, for example, to detect the target in a diagnostic, maybe detecting a marker in blood or urine as a diagnostic. Or if that binding blocks the function of that target, you've got the potential to make a therapeutic and a new drug. Okay, so but the problem with antibodies are they're big, they're complex, they're relatively difficult to develop, they're relatively difficult to manufacture, they're fairly easily destroyed by various environmental and other conditions. And what we have developed in the bottom right-hand corner, and these diagrams are actually to scale, is a small, robust, easily manufactured, easily developed, easily modified to suit the application alternative to antibodies. And as we go through, what I hope I'll be able to demonstrate to you is that it not only operates in those research and diagnostic markets, but also has the potential to work very effectively in the therapeutic side as well. So what is an Affirmer precisely? It is a protein in the same way an antibody is a protein. It's based on a naturally occurring protein, in fact, a human protein. So in terms of making therapeutics that go into people, ideally you want that based on a human protein to reduce the issues around safety and toxicity. It's small and robust. It's very easily modified, as I said, to suit the application. And the way we generate a new Affimer to a new target is very different from the way you generate a new antibody. Typically with an antibody, you take the target and you inject it into an animal, such as a sheep or a rabbit or a mouse, and you let the animal's immune system generate antibodies against that target, which you purify from the blood. That's sort of the basic way they're generated. What we do is, it doesn't involve animals, it's an entirely in vitro laboratory technique. So what we actually have is a pot of Affimers, literally a pot of about 10 billion Affimers, all of which have slightly different binding surfaces that are completely random. We have no idea what they bind to. But when we have a new target that we want to generate an Affimer against, we basically go fishing in that pot. So you dip the target into the pot, and any Affimers you pull out of that pot bind to the target, and that's how we generate the new Affimers, entirely in vitro, without the use of animals. And although that's a minor ethical point in some cases, it is very important in terms of the fact that we can control the way we generate the Affimer. We can make it very specific to the target in ways you cannot do if you're relying on an animal's immune system. And finally, I should say that the technology is entirely proprietary to Avacta, so we own the IP across the board in all applications and globally, and we can talk more about that afterwards. So I mentioned there are some very large markets, and across the screen here you see us going from the research markets where there's no regulatory requirement, so you can put a new product into the research market with no regulatory approval, through obviously increasing risk and reward through to the therapeutics, where there is very significant regulatory requirement to launch a new product, but nevertheless the rewards are enormous in the therapeutic market. And the antibody has come to pretty much dominate all of this. There's very little alternative to antibodies out there on the market. In therapeutics, a third of all drugs in development now are monoclonal antibodies, so there's some very significant markets to have a go at. So what is it that makes an Affirmer competitive? How are we going to compete against a well-established technology like the monoclonal antibody? Well, I'll run through this very quickly because we could spend a long time on this slide, but I'll happily expand afterwards. Very quick to develop. Typically takes 10 months to a year to generate a new high-quality monoclonal antibody. It takes 9 or 10 weeks to generate a new Affirmer because of the way we do it in the lab as opposed to using animals. I made the point about animals, but really that is about speed. It's about the range of targets that you can address. For example, you can't put a toxic target into a rabbit and expect to get antibodies against that target because you kill the rabbit. So there are various targets that you simply can't generate antibodies against. One of the key benefits is it's very specific. So when we generate an Affirmer, we can make it bind to the target and nothing else. So no cross-reactivity, no off-target effects. Typically, antibodies aren't absolutely specific, and again, I can discuss that afterwards. But we can generate very specific, which is obviously important for diagnostics and important for therapeutics. They're very small and very stable. So as I said, the types of things we can do, for example, with engineering them to suit particular applications, we can put them into conditions, into different pHs and different solvents to do chemistry with them that you simply can't address with an antibody. So we have a huge range of things we can do, like attach payloads to them or put fluorescence tags on them, all the types of things that people want to do with these types of molecules. And in the bottom left-hand corner, we should not overlook the fact that Affirmers are very easy to produce. And as we talk through the presentation, particularly on the therapeutic side, you'll see that some of the most valuable types of structures you want to make with these molecules become very difficult to manufacture when you're using antibody-based technology. And we are fortunate, I have to say, because a priori we didn't know that it was going to be the case, but we are fortunate that Affirmers, even in those complex structures, are very easy to manufacture, which is a massive plus. Okay, so our business model, our approach is to focus in both of those areas, in the non-therapeutic space, which we term reagents, and build a profitable business in that area where there's less regulatory hurdle, so it's quicker to market. Build a profitable business over the next few years, licensing Affirmers into the diagnostic companies or the research tools companies. So we're not building a route to market for each of these areas, we're licensing into those third parties. And on the therapeutic side, building both a pipeline of our own assets to, of course, ultimately license, but also working with partners, and we'll talk through those partners through the presentation. So we've been able to build a team over the last 18 months, 2 years or so, that can deliver what I'm telling you about. Tony's here as well, so you'll be able to have a conversation with him after the presentation, so I'll just concentrate on the other three. Philippe joined us very recently as we moved into the commercialization phase of the technology. He was the commercial director of Abcam, a company I'm sure you're very familiar with, so brings a huge amount of experience commercializing technologies in this life science space. Matt joined us a couple of years before, who coincidentally actually was the head of R&D at Abcam, and obviously brought the technology through to the point where we began to commercialize last year. And Amrik joined us from GSK, where he was head of formulation, biopharma formulation, so he runs our therapeutic program. So a really strong team with the necessary to deliver in these different areas that we've talked about. Okay, so remember the business model. Non-therapeutic areas and therapeutic areas, I'm going to run through both of those quite quickly. There's a lot on this slide, but it's really to get across the message that there is a very broad range of applications you could apply Affirmers to in the non-therapeutic space. So clearly you have to focus. If you try to do all of this, you're going to do nothing particularly well. Okay, so we've focused in 3 key markets. And again, we'll perhaps use the time afterwards to talk through the detail of this, but these are 3 key markets that are growing rapidly, where there are problems with antibodies in delivering the applications that people want, and where, as you see at the bottom, there are some key benefits that we can demonstrate and do demonstrate of Affirmers. Specificity, you'll see, is on the top of the list there for all of them, and there are particular benefits in these different markets that Affirmers bring that give us a real competitive edge over antibodies. So Philippe is building a strong pipeline. I think we'll see in a moment, and very recently we signed a deal which was announced on Monday actually, which is probably responsible for the price increase over the last few days, a first diagnostic license to get Affirmers into a very significant top 3 global diagnostic company, and that was obviously a major step forward for us. Okay, so a key message I do want to get across is that we are well beyond the point of asking the question, does the Affirmer technology work or not? We're well beyond that point. We've made hundreds of Affirmers to many, many targets. These performance parameters I can discuss later, but suffice it to say, it hits all the right buttons in terms of performance compared to antibodies. So we are well beyond the point of asking, do they work or not? It's now about commercialization within the different markets that we're discussing. And just to give you a very brief overview of where we're at from a commercialization perspective in the non-therapeutic side, we've been commercializing for about a year, so it's very early stage. But we're now, and these figures have changed over the last few weeks, we're now in more than 4 of the top 10 global pharma, many of the biotech and pharma evaluating the technology. I mentioned there several diagnostic companies, including one of the top 3. And I guess the stop press is that we signed a deal which was announced on Monday with that company. Very limited as to what we can say about that deal, but it's a significant market that they're interested in taking. They've taken exclusivity to several Affirmers that they've evaluated, meet their requirements, and they now go into development. And financially, although this is not a revenue story at the moment, this is very much an R&D-led growth story, but financially we're well on track for this year to hit the market expectations, so no sort of unpleasant surprises. Okay, so let me move on to therapeutics, remembering that business model. I suppose another point that I'd like to get across is that I think it's entirely reasonable to say now that the stage we've got to, the current market cap, 60, 70 million, is entirely supported, I think, by the reagents business. Okay, so a profitable reagents business would be valued at more than that in my view. So the therapeutics piece, I don't think, is priced in particularly to the current share price. So it's a really exciting, higher risk of course, but really exciting cherry on the cake as far as we're concerned. So first of all, again, we talked through the competitive advantages. Why is an Affimer therapeutic going to be any better than a monoclonal antibody therapeutic? Why are we going to be able to compete in this area? And without going into too much technical detail, the 2 technical benefits that are very relevant in therapeutics are around making what we call multimers, so effectively sticking Affimers together to be able to hit more than one drug target at once. That's very difficult to do with antibody technology. And also the fact that we have a small, very simple, easily produced protein. So that leads us through into 2 different areas. The fact that we can make multimers lets us look at what are called T-cell recruiters and agonists. I'm not going to go into the detail here, but very happy to talk about it afterwards. I'll just expand a little bit on T-cell recruitment, just out of interest to give you an insight into what that is. But T-cell recruitment and agonists require you to make these multimer structures. Very difficult to do with antibodies, straightforward with Affimers. Small, stable proteins allow you to think about making drug conjugates, about gene delivery, and about something called CAR-T therapies. There isn't time today, but happy to talk about it afterwards. Our in-house program, so we were funded back in 2015. We raised about £22 million for this therapeutic program around T-cell recruiters and agonists. That's where we're investing our money. And on the right there, the drug conjugates, the gene delivery, and the CAR-T therapy we're doing with partners. So Glythera is a UK biotech with a very novel technology around drug conjugates. Moderna is a hugely well-funded US biotech that is focused on gene delivery with a very novel mRNA technology. And Sloan Kettering we're working with on CAR T. Sloan Kettering is the biggest cancer center in the US, and they were the institute that founded Juno, which is one of the leading companies in CAR T. So on the right, those are all being done with partners. On the left, that's our in-house program. Okay, so I said I'd just say a little bit more about T-cell recruitment because and I think it emphasizes how good Affirmers are and how much of a differentiated product we can bring to the market. So bear with me and I'll just talk you through T-cell recruitment. What you're trying to do with T-cell recruitment is get the T-cell, which is the immune cell, which destroys foreign cells in the body. You're trying to get it and bring it to the tumor cell. Effectively stick it to the tumor cell and say, right, get on with your job and destroy that tumor cell. Okay, so what has been created in the market to date is something that has two, not Affirmers, but antibody fragments. Two antibody fragments, one that binds the T cell and one that binds the tumor cell. So what happens is that almost like Velcro sticks the T cell to the tumor cell, it destroys the tumor cell, and you get a therapeutic, okay? So that's the real basics of T-cell recruitment. The product that's on the market is called blinatumomab. It is the most expensive oncology therapy on the market, and that is not only, but primarily because it's very difficult to manufacture because it's made of antibody fragments. So one thing that we are now beginning to do is look at making a blinatumomab mimic but using Affirmers, where we know making two Affirmers stuck together is very straightforward. Okay, so that's the first step. But what's a lot more interesting and what would really differentiate Affirmer technology from antibody technology is this. When you start to hit a tumor cell with a particular drug against a particular target, and I coloured it purple on the last slide, the tumor cells start to become rich in other targets and start to stop producing the purple target. They evade the technology. It's called tumor cell evasion. So what you'd really like to do is you'd like to target the tumor with two targets, purple one and a green one in this case. So you don't just want a dimer, you want a trimer, you want three things stuck together. Now, that is extremely difficult to do with an antibody. I'll stick my neck out and say that's possibly not even possible to do with an antibody. We've already made trimers of Affirmers. We know we can manufacture them. In fact, we've made tetramers. So the point is here that as the tumor starts to invade your therapy, you start to hit the other tumor cells with the other target, and it's still effective. That's novel. But then the next step is that patients who are given blinatumomab, because it's quite a small molecule, they have to be on a continual pump because otherwise it's cleared through the kidneys very quickly and the time that it's in their bloodstream is very short. So what you'd really like to do is take your system that targets two targets on the tumour, but also add something that piggybacks onto one of the very big proteins in blood so that it can't get cleared through your kidneys. You then have something that stays in the blood for many days, if not weeks, targets two things on the tumour cell and performs a T-cell recruitment. That's extreme, that's never been done. We know we can make tetramers. This is in our in-house pipeline to achieve, and clearly then you can start to provide the market with something that's not only suitable for patients, because they don't have to go into hospital and have a continual infusion, but is also very effective in killing the tumour cells. Okay, so that was a bit of a departure, but useful perhaps to understand how Affirmers can deliver really differentiated medicines that are obviously valuable to large pharma ultimately. Our pipeline looks like this. Talk you through it very quickly. We are building a portfolio of Affirmers that hit many different cancer targets to build those sorts of multimeric structures that we've talked about. We have a lead PDL1 program, program Death Ligand 1 program, which is a major target in oncology. That's quite well advanced preclinically. So we are looking at an IND filing for that program at the back end of 2018 into 2019. So that's when phase 1 clinical trials would start on that lead Affirmer. We also have a program in blood clotting, in coagulation, which I'll talk about later if anybody's interested. And then those 3 collaborations around antibody drug conjugates, CAR-T, and gene delivery with the partners listed there. A question I often get when I give these presentations, or indeed when we talk to large pharma about the potential of Affirmer technology, is toxicity, safety. This technology has not been in human yet, and therefore the obvious question is, does it have some sort of immunogenic response? Could there be a problem with it? It's based on a human protein, so there should not be a problem. But we just announced a couple of weeks ago the first really in-depth study of safety, if you like. Immunogenicity is the proper phrase to use, where you can see on the bottom here we've tested 3 different basic flavors of Affirmer and benchmark that against Avacta, which is a marketed monoclonal therapeutic. And you can see across there's virtually no response to the Affirmers. There's no immunogenic response. There's no safety issue with them. The bar on the right is the positive control. That's what would happen if there was a real immunogenic response. So to put in a nutshell, the data that we published 2 weeks ago and obviously had a very, very positive effect on the share price over the last couple of weeks. The data we presented shows that Affirmers are not immunogenic, clean as a whistle, which is obviously very, very good news. If I'd been standing here a year ago, that list of bullet points at the top was what I was telling people we would achieve over the next year. Fortunately, we've been able to deliver pretty much all of that in terms of the animal efficacy data and the immunogenicity data we've just mentioned very briefly. Building out that pipeline of novel Affirmers against the oncology targets, we're going to update the market in Q3. And developing collaborations to deliver those other disease areas we've also been able to achieve over the last year. So it's nice to be able to show some milestones where what we said we were going to do and we've actually done. Over the next year, I won't read through them all, but there are many good pieces of news flow to come over the next year, next 18 months related to the therapeutic program around those collaborations. That'll read out in the next 6 to 9 months. And of course, the ultimate objective, as I said, is an IND filing to go into phase 1 human trials in 2019. The last slide. Now, it's more difficult to put a value on the therapeutic opportunity for this technology. So really, all you can do is start to look at comparators. And there are some comparators. There are only really 2, I would say, sensible comparators in terms of non-antibody technologies, and those are the technologies out of a company called Molecular Partners, which is listed on the Swiss Stock Exchange, and Ablinks, which is listed on the Danish Stock Exchange. And those are obviously further down the process. They're both in clinical phases. But those are the sorts of valuations that these technologies can attract. And the 2 at the bottom are antibody-based, but the point I'm making here is they're platform technologies. So with Affirmers, we're not talking about making one drug asset. We've got a platform that we can generate many, many assets from. So it's important to make that differentiation. And platforms tend to be ultimately, once you've got several examples into the clinical phase, be valued very highly. So it's really, just to give you some comparators to think about the potential value of the Affirmer technology in therapeutics. So just to summarize then, hopefully what I've been able to show you is we've got a really disruptive technology that can be applied in some very large markets. And we've got a balance between what I would describe as relatively low risk now, in fact, very low risk on the reagents and diagnostics side, where it's all about commercial execution, and we've got the people in place to do that, with the potential upside, greater risk, but the potential upside of the therapeutic opportunity. We are well funded. We've got a 16 or 17 million pounds on the balance sheet, which is sufficient to go through the next 2 to 3 years of program that we set out in 2015. And we've got a very good set of institutional shareholders that support us. Thank you.
Questions, anybody? Oh, there's a gentleman just down front and then second row.
A couple of questions. Firstly, has Woodford got an investment in your company?
No, I've never asked him for one.
Can you use these things to block an antibody?
That's a really good question. Yeah, so without digging into real technical detail, yes. And those things are of interest for autoimmune disease, as you can imagine, where antibodies there's starting to be an immune response that there shouldn't be. And we have many examples of generating Affirmers against the functional bit of an antibody. And just for your information, those are called anti-idiotypic binders, and we have several examples that we can point to. So yes, you can generate Affirmers against antibodies that block the interaction or generally bind to an antibody in its non-functional bit.
Because Woodford's lost a lot of money in Circasa, so maybe you can offer them.
Well, that was the that was the autoimmune side, wasn't it?
Gentleman in the row behind.
I know your diagnostic deal this week, you're very closely controlled by confidentiality over what you can say.
Yeah.
But can you give an idea, for instance, what proportion of your IP estate in diagnostics is it sounds very low.
So just that's
well, you've done a deal that is an exclusive deal in diagnostics.
Yeah.
I don't know if it's 5% of
I
your
understand.
IP or it's over 50% of your IP. I imagine it's quite low.
I understand. Yeah, it's extremely low because exclusivity in that case, and in pretty much all the deals that we do or are doing, we're giving exclusivity for the specific Affimer. So we're not giving exclusivity for the Affimer technology in a particular area. That would be very expensive for someone to get their hands on. But once we've generated an Affimer that binds to their target, and in this case we generated several, because we know exactly what the sequence of that Affimer is, we can license that particular Affimer. So what they've got is the exclusivity for those Affimers against their target in their application. So it doesn't give away a great deal of exclusivity at all, in fact.
You know, it's just interesting that you talk about reagents and diagnostics as perhaps accounting for your full market cap at the moment. With this deal, your market cap's gone up 20-25%, about 15 million quid. If it's a very low proportion of your diagnostics estate, clearly the circle's not squared.
Well, it's an interesting extrapolation, isn't it? Difficult to get those numbers right at the moment, I would imagine. My view is that on the non-therapeutic side, there's a range of deals that we can do. The diagnostic ones are the high-value ones. There's no doubt about that. There's the sort of research tools where you might be licensing an Affimer for someone who makes a laboratory kit to do a particular experiment. Those are much lower value, but you'd be expecting to do many more of them. So putting a total value on all of that at the moment, I think it's too early to do that. So very difficult to put a figure on what the total value of all of that licensing opportunity is. But I would say it easily supports a 50 million market cap.
Any more questions? Gentleman back.
You've given us the upside on Affirmers. What's the downside compared with antibodies? And the second question is, you mentioned 3 global diagnostic companies. Can you name these 3 companies, please?
Yes, that's easy. I mean, they're just by size, Roche, Abbott and Siemens. Sorry, I didn't fully catch all of the first part of the question.
The first question was, you've given us the upside of Affirmers against antibodies. What is the downside?
Okay. Yeah, no, I always try to give a sort of balanced picture. I mean, there are some areas in which you would want an antibody. Again, without going into massive technical detail, on the therapeutic side, what an antibody's function is to identify something, then engage the rest of the immune system. And parts of the antibody that do that, they're not present in an Affimer. So if that was specifically what you wanted to do, you wouldn't choose to do it with an Affimer. Our approach when we go out to do business development is absolutely not to say antibodies are crap and there has been and Affimers are going to replace them, because that would be wrong and very naive. Our approach is to look at those gaps where we can really differentiate what Affimers can do, and I hope I got that across both in therapeutics and non-therapeutics. So really, we see Affirmer technology as entirely complementary to antibodies. And we wouldn't, for example, go and look at Abcam's list of antibodies and pick the top 10 sellers and try and make an Affirmer that did exactly the same. I don't think you'd be successful, because if someone has an effective antibody, they are not going to swap. There's no reason to, and they'd have to revalidate everything they do. So complementary rather than sort of head-to-head competitive.
Time for one more question. Just wait for the microphone if you could. Thank you.
Thanks for your presentation, Alastair. You said you were seeking an IND by the end of this year. Would that be
2018, end of next year.
And next, would that be for one particular cancer or a general set, or?
Another good question. So we have a range of particular cancers that we're looking at filing for. I think we will probably adopt the same approach that most small biotechs do and minimise risks. So it would probably be melanoma, but we haven't absolutely decided that yet, and we will decide that and be more clear about that in the autumn of this year.
And just to add on to that, you're sufficiently well resourced to enter phase 1, assuming all goes well.
Correct,
It does,
yeah.
And then possibly phase 2, but then you need...
No, I would not say we're sufficiently funded to go into phase 2.
I've got a final question. Your share price is up another 8% today. Yes, it's great news. It looks like some is stake building behind the scenes. Can you comment on that?
Not directly, obviously. I mean, we as a company, I've done a lot of work over the last 6-8 weeks trying to get those new significant stakeholders involved who are prepared to buy in the market, and I think we've done that. I think we've been fortunate, but on the back of a lot of hard work, that the results of the half year and the immunogenicity data and the deal this week have obviously been very positive, and that's stimulated quite a considerable demand.
So do you think it's institutions beginning to get the story now and coming in?
I believe and I hope it's a mix of institution and retail demand, but it's certainly having the effect.
Good. Thank you very much.
My
Thank you,
pleasure.
Alastair. Thank you.